So I'm gonna quickly recap mutations for you, and this is better with just audio. Video is kind of useless. I don't put anything up on screen. But yeah, all right, let's get into it. So mutations. Here's some basics about mutations. So mutations in general are any change in the sequence of base pairs of a gene that any changes in the sequence of base pairs of a gene is called a mutation. So it's any change at all in the sequence of base pairs. So some causes of this are any errors during replication or by environmental factors. These are called mutagens. And some examples of mutagens, again, mutagens are the environmental factors, so they aren't actually just like an internal thing, are radiation, chemicals, heavy metals, and viruses. So here's some examples of each one of those. For radiation, X-rays and UV rays are very common. Chemicals, you have asbestos and urethane. Heavy metals, you have lead and mercury. Viruses, you have HPV and hepatitis B. So let's talk about single gene mutations, as that's one of the kind of mutations. So single gene mutations affects only a single gene on the chromosome. So some mutations of this are cystic fibrosis, color blindness, and hemophilia. So two types of single gene mutations are point mutations and frameshift mutations. So point mutations are a single nucleotide base which is substituted for another. For example, let's say we have ATC-CGA, and then it's switched with ATG, so instead of ATC, it's ATG, and then CGA. So this is a point mutation where one thing changes. The next is a frameshift mutation, which is caused by the addition or deletion of one or more nucleotide bases. So this shifts all the future nucleotide bases and changes the amino acids. So that's a pretty big issue. So let's say you have 5, right? And then you read these 3, that's the first codon, and then this isn't a codon yet. But then an extra one gets added in. Then you read these 3, and then you read these 3 as a codon, even though that's not meant to be read as a codon. And then the third one, which would have been with these 2, is now its own first one. So this literally destroys your protein, and it's really not optimal. You don't want this. So there's some results of point mutations, and they all have varying issues, right? So the first one is a silent mutation. So you have to learn about the square, right? And this tells you where everything is. So you have your, let's say, ATC, right? And then that would actually result in AUC because there's no T on mRNA. But if you line that all up, you'd result in some kind of amino acid, right? But not all things are listed to different amino acids. There's 64 things on the square, and there's only 20 amino acids. 11 of them are from your diet, and 9 your body produces, I think. Or it could be the other way. 9 your body, or 11 your body produces, 9 you eat. I don't really remember. But the idea is, it could just still bring the same amino acid, depending on which one got changed. Next is the missense mutation. This produces change in amino acids and proteins. This may or may not change the function of the protein. So the idea is that the amino acid could change, and if it's just a single point where one thing just changes, so a point mutation and not a frameshift mutation. Again, we're talking about point mutations anyways. So it'll change out the amino acid, and in theory that one amino acid shouldn't change that much about your protein in general, so you should be okay. But what if you had a stop codon? You got your AUG. What if you got hit by a stop codon? Then that would definitely ruin it, and that would lead up to the other one, which is the nonsense mutation. This produces a stop codon in the middle of the mRNA, and this will produce a non-functional protein. Not guaranteed, but almost definitely it'll be a non-functional protein. So let's talk about chromosomal mutations. So this is another kind of mutations, and it affects multiple genes on a chromosome, or even an entire chromosome. So single gene mutations affected only a single gene on a chromosome, whereas this one affects a whole bunch of them, right? So some examples of this include Down syndrome and Turner syndrome. So in Down syndrome, one gamete receives 2 copies of chromosome 21 specifically, resulting in the newborn having a total of 3 copies of chromosome 21. This is the characteristic of Down syndrome. Next is Turner syndrome. And one of the two X chromosomes in females is either missing or incomplete. And this also causes some issues, right? So chromosomal mutations can occur when when you change the structure of whole chromosomes as they alter multiple genes on the same chromosome or the entire chromosomal structure. So there's 4 kinds of these mutations. There's deletion, translocations, duplications, and inversions. So deletions is this removes a chromosome segment, right? So a whole segment of the chromosome just gets ripped out and is gone. The second one is translocation, where a segment from a chromosome is transferred to another chromosome. So for some reason, one chromosome has its data, then part of it just gets ripped off and just implanted on the other one. So that's a bit awkward, right? Next is duplications. So a segment from one chromosome is transferred to its homologous chromosome, giving it a duplicate of some genes. So there's an error, right? And then the thing that's copied over just literally gives 2 of the exact same gene. And then inversions. A segment of a chromosome is inverted. So let's say you got ATCCGA. You can probably change up your TCCG and that would get flipped. And then you'd result in AGCCTA, which is not what you had from the start. And this is something that you really don't want. Let's talk about some mutation effects. So gene mutations may produce proteins that are beneficial or harmful to the organism, or they could have no effect at all. And that's your 3 kinds, right? So you have your positive mutation, first of all. This is when a mutation benefits the individual. So, for example, some plants are given resistance to bacterial and fungal infections. A negative mutation is when a gene mutation harms the individual. So some humans have what's called sickle cell disease. So some humans have what's called sickle cell disease, which makes it so that your cells are abnormally shaped, causing blood issues. And then a neutral mutation is when a gene mutation has no effect on the individual. So an example of this is the white Kermode bear. So it's a black bear, but it's changed to a white bear. It still is a black bear fundamentally, but its skin and whatnot is white now. And then the idea of that is it doesn't really change much, right? Of course, they're easier to spot in nature, so they'll get poached and they'll get killed more often. But for the most part, not much of an issue, right? So those are your 3 kinds of mutations. And that's just a quick overview of mutations, and if you want this, I'll have it linked in the description below.